My name is Mikkel Mayer Andersen, and I'm at Aalborg University in Denmark, and it's a great honor to chair this webinar. And a few sort of housekeeping rules first that I want to tell you. So first of all, I'm delighted to welcome fellows and visitors to this journal webinar. Today we will be discussing the paper analysis of forensic DNA mixtures with artifacts by Robert Cowell and colleagues. The presentation will be by Robert Cowell. The meeting welcomes an online audience. Please keep microphones muted and cameras turned off unless called to speak during the open session later on. A recording of this meeting will be posted on the RSS YouTube channel. If anyone prefers not to be filmed, please let us know and we can edit that part out. And if you do experience technical problems online, please use the support chat box and someone in our support team will help you. And we actively encourage everybody to contribute to the discussion of the paper in a Q&A session later on. Comments and questions can be posted in the chat. You're also welcome to raise your hand. So a presentation of the paper by Robert Cowell. The paper will be presented by Robert. It was read to RSS in June 2014 and was published with discussion and replies in the Journal of Royal Statistical Society, Series C, in 2015. And I'd now like to call Robert to present this paper. Thank you, Robert.
Thank you very much. I'm very happy to be here today. I'd like to thank the RSS for the opportunity to talk about the paper. The paper describes a model for analyzing DNA mixtures, and I will start with explaining a little bit of the terminology of DNA for this before discussing the model. I need to share the screen. Let's get my slides up. How do I do this? Can you see my slides now?
We can, yeah.
On full screen. Okay.
Perfect. Thanks.
Yeah. So the paper itself gave quite a sort of long discussion in the introduction about explaining the terminology. I don't have time to do that, so I'm going to give a very bare bones description of the data we're going to look at with the model by reference to a criminal case that was used in the paper dating from 1998, or the paper from there. This involves the genetic profiles of 3 individuals in a criminal case. K1 was a dead victim, K3 the defendant, and K2 another person. And there are 2 bloodstains recovered at the crime scene, and the forensic scientist believed from their examinations that all 3 people's DNA were in the bloodstains. You'll find references sort of at the bottom slides as I go along. So we have 2 types of data. We've got qualitative data on the profiles of the people and quantitative data, peak heights from the DNA stains. The data on individuals are genotypes. These are unordered pairs of alleles on each of 10 different loci or markers. A locus is a position on a chromosome where an allele can be found and measured. Measured is a length, and an allele can have between 10 to 30 length variants depending on the locus in question. And the allele's length is used to identify it, so 2 alleles of the same length are identical, and they're measured in units called repeat units. The alleles in a pair constitute the genotype of the locus. They may differ in lengths, and taken over all loci, that is what the profile of an individual is. For the 2 bloodstains, we get quantitative data, which is a set of peak heights for every allele variant of every locus. And the way these are obtained, in very short description, is you select a small number of cells from each stain, about 100 or so, containing chromosomes. You extract the chromosomes and use a process called the polymerase chain reaction to make billions of copies of these alleles that you're after. You then measure how much of the amplified product there is. And this is found from reading an electropherogram. If you look at the paper on figure 1, there's an example of an electropherogram. and these are expressed as peak heights. So the larger the peak height, the more of that type of allele there is in the amplified product. Now, the artifacts of the paper refer to various complications around these peak heights when measured. One artifact is dropout. So you've got assumptions about who is in the mixture, so you might expect to see a peak at various places, and sometimes you don't see a peak. That is called a dropout, and it's common when the amount of DNA from a contributor is very low. The sort of converse of that is when you see extra peaks which are not expected. And there are three main DNA explanations that could occur here. You might have untyped contributors to the mixture. There might be some sort of DNA contamination of the sample when it's processed in the laboratory. This is called drop-in. A very common explanation is stutter. When an allele is copied during the amplification process, sometimes it's miscopied into the variant one repeat shorter. And these miscopies show up as an extra, typically small peak next to a big peak where the allele originally came from. There's also non-DNA peaks. If you look at the electropherogram, there's usually lots of small peaks, not of DNA origin. And these are some sort of instrumental noise to do with the apparatus that's measuring. I have no idea exactly what this noise is, but it's there. So these are the artifacts that we need to consider which complicate the analysis. So we have the data, the genotypes and peak heights. What do we want to do with them? In this case, we want to know, is the defendant have their DNA in the sample? And this is carried out by calculating something called the weight of evidence. We formulate two hypotheses. The prosecution hypothesis says this set of people is in the mixture, and in those people you include K3, for example. And the defense says, no, it's not those people, it's everyone but K3. It's not K3, it's some random person from the population. Now, given those two hypotheses, we calculate the likelihoods based on the peak heights we've observed and calculate the likelihood ratio. We take the base 10 logarithm, and this number is called the weight of evidence, measured in units of bands. So one band is 1 unit on the log base 10 scale. Weight of evidence, if it's positive, is evidence in favour of the prosecution. The greater the weight of evidence, the stronger the prosecution's case. And the paper shows that there is an upper bound to the weight of evidence. It can't increase indefinitely. And it's given by the negative log of the match probability. The match probability is a probability if you take a person from random population that have the same genotype as, in this case, K3. It's also shown in the paper that the ratio of the maximized likelihoods under each hypothesis also obey this inequality. So what we're after is to calculate these likelihoods, given the genetic profiles on the individuals and the mixtures. So the statistical model we use for doing this is a joint model. It combines one for the genetic probabilities, the genotype of the contributors, and another for the peak height likelihoods given the genotypes of contributors. And there's a nice separation between the two. The genotype model is possibly the simplest you can have. It assumes you have a population in Hardy-Weinberg equilibrium. What this means is if you look at a single locus, you take a random person from the population, then the probability of seeing a particular genotype is one of these two forms. It's the first type, if the two alleles in the genotype are the same, and it's the second type if they're not the same. The allele probabilities here, the PA, are assumed to be known and fixed for each locus. There's another set of assumptions in the model which, rereading the paper, do not seem to be explicitly stated, although they are used in all the calculations. And these are various independence assumptions on the profiles of the individuals. First is that the genotypes of the untyped people, is independent of the known profiles. So it boils down to a joint distribution here. Furthermore, this joint distribution factorizes as a joint distribution over the individual loci as a product. And then further, each of these locus-specific joint distributions factorizes to marginal probabilities for each. So it's a nice sort of factorization there, which we make use of. So now I can talk about the model distribution for the peak heights. I'm doing it slightly different compared to how it's presented in the paper. So we start off assuming that they have a DNA mixture in which the genotypes of all the contributors are known. We look at a single locus, which keeps the notation a bit simpler. Then the total amount of DNA coming from individual I of allele type A from that locus is proportional to the product of three terms. The first term is the rho, which is proportional to the total amount of DNA from all the contributors. This is a single parameter, it's common across all the markers. The second is the proportion of DNA from individual I. That again is common across all markers, and it has constraints that are non-negative, and they sum to 1 when you sum over all the individuals. Third number gives the number of alleles of type A in the genotype of individual I. Note that because the genotype consists of unordered pairs of alleles, we have the constraint that when we sum this number over all possible alleles, we get value 2. Now, we assume that this DNA amplifies to produce both starter and main peaks with independent gamma distributed peak heights of the following form. And you can interpret this as saying a certain fraction, C times the amount, goes into producing the stutter, indicated by the superscript here, and the remainder goes into producing the main peak at A. We assume these are independent gamma distributed random variables with a common scale parameter. And this is the same for all the alleles and all the individuals. So eta is a common scale parameter. So to get the total peak height, we add these for each allele over all of the individuals. So we take the sum of the peak heights on allele A from all the individuals, the main peaks, and then we get a contribution to the peak at A from stutters arising from alleles at A plus 1. So we have this secondary term. And because everything all independent have the same scale parameter, the result is something which has a gamma distribution of this nice simple form. So that is our model for incorporating stutters. We need to now incorporate the artifacts of the title of the paper. So drop-in is modeled by adding enough extra untyped people as the sources of the drop-in peaks when you formulate the hypotheses. It's very common to specify a certain height C, called the analytic threshold, in which all peaks below C are ignored. So we do this, and any peak below the threshold C we treat as having a 0 height. So this models both dropout and noise. So essentially, if you do see a peak which is positive but below C, it may have come from the allelic material, but we treat it purely as noise and say there's been a complete dropout of that allele there. So what we observed are either values of 0 for peaks below the threshold, or the actual peak if it's at or above the threshold. We give to the 0 peaks the probability computed from the cumulative distribution function of the gamma distribution. The peak height likelihood, remember we've assumed we know all of the genotypes of the contributors. The genotypes, given the likelihood, factorizes into a product of likelihoods over all the different allele variants of each marker. So our Zs are the observations, as said, all possible peak heights of all allele variants of the alleles over all the markers. Each term here is going to be either calculated from the gamma density or the cumulative distribution function, depending on the value of that particular peak of that allele. Now, to get the overall likelihood, we need to average this using the probability distribution of the untyped contributors. So we form this sum here. So n here represents all of the untyped contributors. For brevity, I'm not indicating any type contributors here. Make the formula simple. So this is a big sum, but because of the factorization we have of the genotype probability over the different markers and the double factorization we have this L term given the genotype, we can rewrite this big sum as a product over markers of the joint genotype probability on the marker given the hypothesis and the likelihoods for that marker. We want to maximize this whole product by varying these parameters. So there are not very many parameters to maximize. We've got 3 scalar parameters. We've got this phi, which has the number of phi's we have is a number of unknowns, but we have the constraint that they add to the number, and the phi's have to add to 1, so that reduces the number of independent variables. Now, one issue here is that the number of terms here can grow very, very large with just a few number of contributors, and we'll see by some numerical examples how big that can be. So we want to take care of that. So, I mean, it's shown in the paper how to do this with Bayesian networks. So this is one Bayesian network which could be written down, but it's incorrect. I emphasize it's incorrect. The nodes set here are our peak heights. We have 2 contributors indicated by 2 sets of n nodes, ni and nj, and 6 alleles on this locus. So the network is indicating the correct dependence of each peak height on the allele and the allele 1 up, which stutters to it. But it's incorrect because, as written, it implies that all of these n nodes are independent. But that ignores the constraint that they have to sum to 2. Each of them has to sum to 2. So the paper shows how this can be circumvented by introducing another set of nodes, noted by S, also having 3 possible values. And these are constraint nodes. So within the Bayesian network, you give them probability tables. The probabilities take values either 0 or 1, depending on whether the constraint is obeyed or not. And the constraints we have, we start off with n1 takes a value s1 is equal to that, and s2 is equal to s1 plus n2, and so on. We go down the chain, please. And finally, we have the last one has a constraint that's equal to 2, and that's the sum of the NIAs. And similarly for the NJs. We also have the constraint that the only allowed values for NIA, each term here, this cannot be bigger than 2 minus the preceding cumulative sum. So any values of n which violate this constraint are given 0 probability in their probability tables. So in this way, we can obey the constraint. It's shown in the paper how to give binomial distributions to these NI nodes, such that the Bayesian network will contain the Hardy-Weinberg probabilities that we want. Now, one of the nice things about putting it in the Bayesian network is that we can use very efficient algorithms now to calculate the likelihood, which have been developed for Bayesian networks. I can't talk about those algorithms, but I just will concentrate on the complexity of the algorithm, the calculation involved constructed what's called a junction tree from the Bayesian network. And this consists of cliques and separators, groups of nodes of the table, put into a hypergraph type structure. For this Bayesian network, the cliques can be chosen as all the nodes in two consecutive columns, and the separators, all the nodes in each internal column. And we associate a joint table with all these nodes, all these cliques and all these separators, and all the calculations can be done with these quite small tables. Now, if we look at the separator, that involves one S and one N node. So there's 9 states there altogether, but taking into account logical constraints, this reduces to at most 6 possible values. Cliques have 4 such nodes, gives you 81 possible states, but at most 10 can be non-zero. That's a much bigger reduction. In general, if we have A alleles and a locus, we'll have A-1 cliques and A-2 separators. And if we have U-type persons, each clique will need 10 to the U values and each separator 6 to the U values. So we add these together, and that gives us the total size of all the tables we need to do the calculations to find the likelihood. We can compare that to the joint genotype space, which is order A to the power 2U. So I've given a couple of examples here. For if A equals 14 and U, we have 4 unknowns. The joint genotype space has about 10 to the 8 possible values, but only less than 146,000 can be non-zero at most. So that's a big reduction, a factor of over 800. If we have 20 alleles and 5 unknowns, we need just over 2 million numbers to be stored for the clique separators, but over 10 to the 11 for total joint genotype space. That's a huge saving of over a factor of 200,000. So these big savings allow us to calculate likelihoods using the Bayesian networks much more efficiently than doing a brute force calculation going through all the possible genotypes individually. Now, there are other aspects discussed in the paper, which include various things, detailed analysis of the crime data, mixture deconvolution, where if you have unknown people in the genotype, you want to infer what their phenotypes are, given the peak height data. So as I combine one or more samples of replicates into Bayesian networks to calculate silent alleles, so on. Very many things, don't have time. I should mention that the DNA packages by one of the co-authors was used in doing all the computations of the paper. This is open source, but does rely on the Huygens software. It can be downloaded from this link. And for even more explanations of the use of the Bayesian networks, see the second paper here. Now I want to talk a little bit about extending the model, which can be done on 2 independent fronts. You could change the probability model for the genotype, you could change the peak height model. If we look at the genotype model, we've assumed allele frequencies precisely known, Hardy-Weinberg equilibrium, and independence of contributor genotypes, which essentially treats all individuals as unrelated. All of these assumptions can be relaxed, and Peter Green will talk about that after me. I want to discuss one extension, which is the inclusion of forward and double reverse stutters. A forward stutter is when, during the amplification process, a miscopy happens and an allele 1 unit longer is created. Double reverse stutter is miscopy where 2 repeats are dropped. The way to model that is straightforward. We now need 3 types of stutter parameters, which are 3 types of stutters. And we assume that all of these stutters are independent, gamma distributed with common scale parameter. Then when we add these all together over all the individuals, each peak will have a gamma distribution of this form where now we have modified our dA function. So that's the model side. This is easily incorporated into the Bayesian network. I've illustrated this just for one node, Z3. And if I put in red arrows everywhere, it'll look a bit messy. But we can see we have the correct dependence. The arrow coming from N2 gives you the forward stutter, and the arrow coming from N5 gives you the double reverse stutter. We do this for both contributors. But now our cliques now have spanned 4 consecutive columns, and the separators span 3 consecutive columns. So this increases the complexity. So the question is, by how much? And apparently quite a bit. Separator, we have 3 NS pairs. There are 729 possibilities between them. But when you apply the logical constraints from the S and N nodes, that reduces to at most 15 possible non-zero values. For a clique spanning 4 columns, we have 6,561 possible states, but at most 21 can be non-zero. That's a huge reduction. So we take a clique with 4 untyped persons. That'll be this number to the power 4. This is almost 10 to the power 16. So in some context, to store that on your computer, you'll need about 14 million gigabytes of data of RAM. On the other hand, 21 to the 4th is only slightly less than 200,000, requires just 1.5 megabytes of data. So because of this significant reduction, we're able to extend the model to both forward and double reverse stutters. And you can see by the small numbers here that we could possibly go even further. So when I was a lecturer, I used to give small exercises in my classes. So I'll give one here. So we're seeing the sequence of reduction 6, 10, 15, and 21. Can you work out what the next number will be? So when I was asked to give this talk, I was asked to talk about the impact of the paper. I see that on Google Scholar, there's over 130 citations to the paper. But rather than talk about the academic impact, I want to talk about what I think is possibly one of the biggest impacts, practical impact, and that's the development of this software called Euroformics. Soon after the paper was published, Peter Gill initiated an implementation of the model in his group in Norway. The original software was tested against the calculations of the paper and verified. That's a good validation of the model. Since then, the Euroformics model has been extensively developed and is used worldwide in forensic laboratories. And I've seen a recent paper where validation studies being carried out in Brazil for its use in Brazil. It's fast. Didn't realize how fast it was nowadays. Peter Gill sent me some timings. For 4 unknowns, it takes about a minute. And for 5 unknowns, about 15 minutes. You know, timings all depend on particular mixtures, how nice they are in some ways. There's also another paper. Sorry, you can download and see all the extensions on this from this web page. There's also another package developed by the NFI, which implements a model similar to Euroformics and understand the 2 groups cooperate to check each other's software as given the same answers on same sort of problems. So I want to now finish on 2 aspects of mixtures that I've been working on the last few years. The first is a quite large paper I put out about 7 years ago. It's a different mixture model which uses probability generating functions to model the peak height distribution. It uses multivariate probability generating functions and work that I.J. Good did on cascade processes from about the late 1940s, early 1950s. what I show is that you can actually calculate in full these probability generating functions numerically very efficiently by using fast Fourier transforms. In the simplest case, it only takes a few lines of code in R. So if you've not seen this paper, you might be very interested in to have a look at it. I do warn you, this archive paper is very long, but it does have lots of tables. It's not as long as it seems, but about 180 pages. But I think another reason I want to mention is I think the idea of evaluating the PGFs with fast Fourier transforms should be applicable to other fields beyond forensic DNA analysis. So, for example, spread of disease in epidemics and so on. Also, if you're teaching students, I think it could be a good source of topics for final year project students, for third-year-old master's students. More recently, I've been working, for the last couple of years, I've been working what I think is a new paradigm for analyzing mixtures. It's a single cell analysis. So I was invited to join a small group a couple of years ago, and this group has a large data set of about 3,000 single cell EPGs. So the thing is that technology nowadays is so advanced that you can automatically isolate and amplify individual cells in a DNA mixture. So if you apply this technology, you get from a mixture, you get a large set of individual single cell EPGs. So each cell just comes from one person. So ideally, you could take each such EPG, electropherogram, and read off the genotype of the person it came from by identifying the one or two largest peaks on each locus. Unfortunately, things are not so simple. In practice, we have very high dropout rates, typically over 50% of alleles and loci drop out. There's much greater peak height variability compared with standard mixtures, and starter peaks tend to be larger relative to main peaks than in standard mixtures as well, sometimes even exceeding the main peaks in heights. But suppose you have a set of such EPGs, then the computational task is very different compared with the standard mixture software. The initial task is to partition the EPGs into subsets, such that all cells from a contributor are in a single subset, and all cells in a single subset come from a single contributor. There's one such partition for which that is true. Having found that partition, you can use it for likelihood evaluations, like ratio evaluations, and also use individual subsets for deconvolutions if you don't know the genotype contributor to that set of cells. The advantages, as I see it, over current mixture analysis is that there's no DNA amount to estimate. We know we have one cell per EPG coming from exactly one person. It also means we don't have allele masking arising from two people sharing the same allele. The number of contributors of a mixture arises as an output. It's the number of subsets in the partition. It's not required as an input, and sometimes that's contentious in standard mixture analysis. How do you choose how many contributors to specify in your hypothesis? The deconvolution of multiple untyped contributors, you don't require the amounts each contributes to be quite unbalanced to distinguish between them. In standard mixture analysis, you tell different contributors apart if they contribute very different amounts of DNA. One person will have high peaks, the other low peaks, and this is how you find the genotype. But you don't have this issue with single cell analysis. But there is a but. We need to find the correct partition. If you have 100 cells, the number of partitions is given by the 100th Bell number, which, as you see, is a very, very large number. So an exhaustive search is out of the question. So we need to do some heuristic searching. So I have a paper coming out, which I'll put on the archive saver soon, details down there, in which I present greedy search algorithms for finding very high likelihood partitions. Having found a set of partitions, we can take the highest likelihood as the maximum likelihood estimate of the true partition, or the set of retained partitions for Bayesian analysis, assuming that one of the retained ones is a true partition. The paper shows through many simulations using these real lab-prepared single-cell EPGs, it's both efficient and accurate. For this talk, I've done another small simulation to illustrate this. I simulated 5 mixtures, each of 100 cells. Each mixture consists of 10 cells from each of 10 contributors, each treated as untyped. So I got 5 mixtures, each of 10 unknown people. It's all perfectly balanced. To greedy search with partition likelihoods, which I evaluate using the discrete cell model of the previous archive paper I gave the details about. When doing the search, I mentioned there's a great deal of dropout. So I discard cells which have high amount of dropout, so very low quality, they can't tell you too much. But that still meant that between 75 and 85 cells were retained in each simulation. 75th Bell number is about 10 to the 80. In all 5 simulations, the highest likelihood partition found by the greedy search was found to be the correct partition. I already know who the true contributors are to each cell, so I can check this. So I got the true partition. And each of these partitions was found in about 25 seconds on my 9-year-old laptop. So it's very fast, very efficient, and looks to be doing things that standard mixture analysis can't do. If you want to read more about single cell analysis, I was involved in a paper that came out recently, so you can look at this paper and the references inside that to follow up on the ideas. Thank you very much for listening, and here's the answer to the exercise I said.
Thank you very much, Robert. Thanks for a really, really good and interesting talk. I would like to pass on the microphone to Peter Green, who has also a presentation and would like to discuss a bit before opening up on the Q&A. Robert, you may need to, yeah, exactly. Now you can share his screen, I hope.
Oh,
excellent. Thank you very much, Peter.
Great.
Is that visible?
Yes.
It is.
Yes, thank you.
So, mute me.
Good afternoon, everyone. Thank you for joining us. And I greatly appreciate the RSS and Anna setting this event up. And I greatly appreciate the work of the 4 authors, which I've known about for a little longer than the life of the paper, since I was in touch with the authors before it was finished. They were very secretive about it, much to my irritation, but it was worth waiting for. So I'm going to talk about the work I've done in many cases, mostly with Julia Mortera, one of the 4 authors, extending this model and the analysis in a few slightly different directions than the ones we've just been hearing about. So just a quick recap, that the whole process by which genotypes lead to an electropherogram representing a mixture of DNA is very, very complicated. But broadly speaking, there are 2 parts. There's the part that leads to the creation of the DNA sample, where multiple individuals' samples are mixed physically into a sample that is found, for example, at a crime scene. And then the process by which that sample is analysed using PCR, as Robert has described. So we've got this simple graphical structure at a global level, and for most purposes, we can collapse that and just think about this rather simple structure here. And it's natural to assume, and we do pretty much throughout, that the process by which this joint model is created is it factorizes it into 2 conditionally independent parts. The process that generates the electropherogram given the genotype numbers, given the genotype profiles, and the process that governs who is in the sample in the first place. Now, this is a wonderful paper to read and has many interesting features that that fit together in a rather harmonious way. We have the gamma peak height model for the EPG. It's a model that captures many, perhaps most, of the important artifacts. It exploits a Bayes net representation for the genotype numbers, and crucially, because the electropherogram is fixed, even though those are, of course, are continuous random variables in a model, because they are fixed, the model for the genotypes given the electropherograms is still a discrete graphical model, and it's still a Bayes net, and that's crucial for the computation. They've got a clever allele count representation, which allows exploiting the Markov serial dependence along the allele values, as Robert's described. And there's this package that Robert's already alluded to called DNA mixtures. And I believe Therese is present today. So thank you, Therese, for that. Now, the starting point for the work I've been thinking about around relatedness among contributors starts with the familiar problem of paternity testing. So we have a typical case, the mother, a child, and a putative father. And we wish to assess the evidence for paternity from these three genotypes. But what if the father's or putative father's genotype is not available? Suppose all we have is a biological sample that appears to be a mixture of several individuals, possibly including the father. Can we still proceed? Well, one simple-minded way to do it is, for example, to analyze the mixture as if it, using this model, using none of the relatedness structure, and then just to test for paternity in the usual way. That's obviously not very satisfactory, rather arbitrary, a sort of plug-in approach. So we want to try and build the relationship and relatedness model into the model itself. Now, in reality, genotypes of the various actors in the scenarios of DNA mixtures are not independent. And there's two kinds of relatedness, and it's important to distinguish them. One I sometimes call the ambient relatedness. There's always a degree of kinship among individuals in subpopulations, and for that we have familiar corrections, the theta correction, for example, which is based on a putative island model for subpopulations. But what we're talking about here are specific close family relationships. Under some of the hypothesized scenarios, some of these actors are close relatives. And paternity cases is one obvious case in point, but there might also be a question of there being brothers or families in a joint criminal enterprise. And in this latter case, we may require analysis about such relationships, where that's the topic of the question. Is this or is this not the father? Or we may require an analysis that allows for such relationships. Was the suspect's brother also at the scene? And the advantage of a full model-based approach to the mixtures, taking into account possible relationships, is that these can be dealt with in a unified way. So what we're going to do is the simplest thing possible, really, is to replace the joint distribution of the genotype contributors. Instead of thinking of this as k independent draws from a gene pool, we're going to model the hypothesized relationships through that replacement model. And we're going to model relationships among the contributors and also between the contributors and other individuals who are typed, whether or not they contribute. Now, the key to this is basically Mendel. The key to this is identity by descent. At the level of precision we're talking about here with this kind of modeling, the sole genesis of relationships is identity by descent. And here's a rather pretty colored diagram stolen from Wikipedia, illustrating the idea of IBD, identity by descent. So here we have the colors represent the different allele values at a particular locus, and we see here how as through these 3 generations, we see in one realization how these alleles are passed down. And in this particular family tree, there's obviously some inbreeding here, because there's a first cousin marriage here. And if we look in this realization, the red allele goes down through the aunt to the cousin to the child, but also through the mum, the other cousin, and the child. So this loop in the pedigree gives rise to the possibility of identity of inbreeding in this individual. So these alleles are representing, so these blobs are representing alleles, and thinking about how these alleles are passed down through the generations is what allows us to unpick relationships. So we're sticking to the standard scenario, autosomal markers, one at a time, Hardy-Weinberg equilibrium, and so on. Think first about a single parent and child. Under this scenario, under this framework, the joint distribution of their genotypes is precisely described as follows. If the parent's genotype is AB and the child's is AC, then A, B, and C are independently drawn from a distribution of the alleles. The distribution depends on the population and the marker and so on. The A appears in both of those genotypes. That's what we mean by saying those genes are identical by descent. In the absence of inbreeding, any 2 relatives' genotypes can be written as AB and either AB, AC, or CD. They're the only possibilities. We don't care about the ordering among the genes here. So either the second relative is identical to the first, or has 1 in common or 2 in common. The probabilities of these are usually represented by kappas, kappa 2, kappa 1, and kappa 0. And the individual values are drawn IID from the gene pool, and the sum of the kappas is 1. So parent and child relationship is summarized by saying kappa 1 is 1, and 2 siblings can be summarized by saying kappa 0 and kappa 2 are a quarter, and kappa 1 is a half. And that completely describes the situation between 2 individuals at a single locus in the absence of inbreeding. When you allow inbreeding, obviously it becomes a slightly more complicated, but we've known how to do this for at least 50 years. And there are some so-called condensed coefficients of descent, usually denoted delta, which summarizes the various patterns of identity you can have among 2 individuals. And now moving on beyond 2 individuals, well, that of course has been studied. Elizabeth Thompson is a source of some very deep work, profound work, very pure mathematical work, mapping all that out. But we wanted to find a rather simple representation of a potentially rather complicated situation. And that gave us the idea of the IBD pattern distribution. So to do this gently, let's think about a father, mother, child triple. Father's genotype 1, 2, mother's genotype 3, 4, child's genotype. Well, the child will have 1 gene from the father, 1 from the mother, and the 4 possibilities are there: 1, 3, 2, 3, 1, 4, and 2, 4. But if we don't care about maternal and paternal genes, and if we don't care about the order of the 2 genes, then all of those 4 patterns are the same. They're saying that assuming the father and the mother are not related, there are 4 independent genes from the parents, and the child has 1 of those from the father and 1 from the mother. And now we can build that process up. We can now think about other relatives in the pedigree. For example, we could now go back a generation to the paternal grandfather, and the father's genotype was 1, 3. So either the 1 or the 3 came from his father, and the 5 came from his mother. And so we can see how we can build up the pattern through the generations. And using these two steps, the idea of condensing, because we don't care about order or distinguishing maternal and paternal, and think about adding new individuals with the simple application of Mendel's law, we can build up very big structures. Now, you may think, well, this is a lot of stuff for a very simple idea. We know how to quantify a relationship between two individuals through kinship coefficients and so on. But it's crucial to note that pairwise relationships do not determine joint ones. So here are two possible pedigrees for three individuals, all cousins of each other, pairwise cousins. They may be what you would expect, that these three children are children of three different mothers who are sisters in a single family.
Peter, sorry to interrupt. I have to take the nasty role of being at the chair now and having you to mind the time.
Okay, thank you very much. Yes, I will roll on. So what we can do, it's important, if you study the IBD pattern completely, you can see that these are really different, and that's important. Now, what we do in my modification called KinMix of the DNA mixtures software is to automate the process by which the Bayesian network that Robert described is modified automatically through reading this table. And here's a particular illustration of the Bayes net, which you never need to look at, of course, the Bayes net that's generated from that pattern. So just to summarize the software that's available using these ideas, Therese's package is there. My package augments it to include these relationship details. They both build on Hugin, as mentioned, which is expensive but fast. We also now have freeware versions of these, entirely independent of commercial software, with some limitations on speed and functionality, but available on CRAN. And you can find out all about these at this website. And just to give you just one illustration, here's a small family where I've got well, you can see what I've got. We've got two individuals who are typed, the child and the maternal grandmother, and we're supposing that the mother and the father are in the mixture, and this allows us to create that model mnemonically, which captures all those relationships. So thanks for your attention. Sorry to run over. There's other work that I could have talked about jointly with Julia, and I suppose these slides will be available afterwards, and you're encouraged to look into some of these ideas offline. Thanks for your attention.
Thank you very much, Peter. So I would like to congratulate Robert and his co-authors with the seminal paper that Robert presented here. And also thanks to both Robert and Peter for the excellent, insightful talks you have given. Thank you very much for that. So now we open up for the Q&A session. I know the time is scarce, but we do have time for a few questions. So I hope people either will put in a question in the Q&A section or raise their hand. So there's I think that's in the chat here from, so John is just saying, thank you very much. Okay, so maybe I can ask a question. So Robert, about this number of contributors, that's always sort of a question. How many should you take, and what is the software able to? What are your takes on that? I mean, you have presented the original paper up to 4, maybe 5 contributors. Yeah. Is that sufficient? Do you think there are some mixtures where more are needed?
I think there are some cases. I remember many years ago, Julia was asked by forensic scientists in America wanting to go up to about 7 people because the group in a gang was sharing balaclavas. So I think that you can get these more extreme mixtures. There's also crime scenes are not usually very clean. So like the bloodstain in that case example was recovered from mirror in the public laboratory. So there's not a clean surface. There'll be lots of other people's DNA in there. So yeah, I think it can happen.
And you in the beginning of your presentation, you had a proof that the random match probability was an upper bound of the weight of evidence. Can you do something similar if you misspecify the number of contributors? So say there are only 4 contributors.
Well, I think it's in the case if the defense hypothesis is the same as the prosecution, but you replace your defendant by a random unknown person. Now, I think the match goes through however many contributors there are.
Okay, thank you. So Peter Gill has raised his hand, so I'll pass the word to him. Hi, Peter.
Hi. Yeah. Just a quick comment really on the contributor issue. Of course, we never know what the number of contributors are. This is an impossible question, and so what we're more interested in is in the effective number of contributors, which is different. So we think of the software as exploratory. So what we would do is, if we weren't sure of the number of contributors, maybe we thought there were 4 contributors, for example, then we would run the software with 4 contributors. And then if you go to 5, 6, or 7 contributors, for example, what happens is that you find that the likelihood ratio will stabilize. And the reason it stabilizes is that if you have too many contributors, the mixture of proportion values become minute. And because the mixture proportion values are so small, they have absolutely no effect on the LR. So I always think of the number of contributors as a red herring, really. It's kind of a nuisance parameter, really, but it's not as difficult as people make out.
Okay, thank you, Peter. So maybe I can ask a question about the parameters, Robert, that you also mentioned. So there's a stutter parameter, for example, and I found whether in the generalization, whether that was the same across all loci. And so what would happen if you had a locus-specific stutter rate? Is there any risk of overfitting or something like that?
Right, I think what you should do, I mean, the paper did talk about locus-specific and I think allele-specific rates. I think they shouldn't be estimated from case to case, but they should be calibrated into the model by external database of cases or laboratory samples, and sort of fixed values used within. That's my feeling the way it should go. Is that in my more recent software, that's the way I go. I don't estimate those, they're input parameters.
Okay. So I have a last question if nobody else will. So what is the current model weaknesses? So in the paper and also in your presentation here, you discussed some, and Peter Green discussed one of them about relatedness between contributors. What is the weaknesses that you need to focus on from a research perspective?
I think for that model, well, I think if you introduce relatedness, I assume that you're still using independence between loci. So perhaps if you have syntenic, is that the word? Loci, the loci is very close on the same chromosome, so you can't assume recombination factor of 1/2. So you get non-independence of loci for related individuals. I think it's an interesting problem.
Well, yeah, but that's separate from the relatedness issue. Surely that issue arises for any
but it only really applies to relatedness. If you have related individuals and the loci are linked, then you can't use the product rule. You have to, in a sense, you have to calculate likelihoods for both loci together. You can't do them separately and multiply them. But if you don't have relationships, then that's not an issue. It's only when you have the relatedness coming in, and also only on some certain pedigrees and not others. I don't know.
I think we'll have to disagree on that one. But
okay, I would like to close on this disagreement, if that's okay, Peter, or unless you have a brief comment.
No, I just want a brief comment on the previous part of the discussion about number of contributors and so on. I think we shouldn't forget this is still at the end statistics. We're talking about parametric statistical models, and we shouldn't forget about all our understanding about lack of identifiability and near identifiability. And we shouldn't expect in noisy data to be able to discriminate very finely different models. And it just becomes particularly difficult with very small contribution probabilities, of course.
Thank you very much for that remark, Peter. That was important. Okay, so I'd like to thank both the audience and of course the presenters for making this webinar possible. That was really, really nice and interesting. About the next journal webinar, some full details of the upcoming journal webinars are available on the RSS website and announced in the members' newsletters and on the RSS LinkedIn page. And if you'd like to receive an invitation for future journal webinars, please email journal@rss.org.uk, and they will be sure to add you to the mailing list. So thank you very much, everybody, for coming, and have a nice day.